We humans have an incredibly sensitive apparatus within our ears that allows us to hear and maintain our balance. However, exposure to loud noise and ototoxic drugs can result in debilitating damage to our hearing and balance organs. My personal interest in regeneration, together with the clinical need for better therapeutics targeting deafness, led me to Jeffrey Corwin's laboratory. 2 decades ago, Jeffrey Corwin and his group discovered that birds regenerate their hearing ability after injury. Birds can regenerate their entire pool of hair cells. Humans, and other mammals for that matter, clearly cannot do this. This led to their publishing a seminal paper which set the stage for inner ear regenerative biology for the next 20 years. Since then, neuroscientists have sought to identify differences in the ears of mammals that might help to account for the absence of the rapid and innate regeneration that leads to hearing and balance recovery in those non-mammals. This is where my project comes into the picture. As part of a distinguished major's thesis in biology, I'm attempting to find those key molecular differences. A former graduate student in the lab, Joe Burns, noticed that the cytoskeleton of supporting cells, those cells which neighbor the hair cells in the inner ear, are unique in mammals, notably mice. He observed that the F-actin belts, a structural difference that helps to keep the cells rigid near their circumference, was unusually thick in adults. Similar experiments in birds, animals that can readily regenerate their hair cells after injury, showed a consistently thin F-actin belt throughout their lifespan. Based on these results, I decided to investigate how these belts get reinforced at the molecular level. There were a number of potential targets, but we decided to look at myosin II. We carried out a series of experiments attempting to figure out the potential role of myosin II in its interaction with F-actin. We used a drug that inhibits the action of myosin II, called blebbistatin. Early investigations with young chicks and adult mice showed remarkable results. This is what a normal sensory epithelium looks like. It's a fairly level surface with hair cells located between supporting cells. When treating with blebbistatin, we see a significant shape change. This tells us that myosin II regulates the tension between the supporting cells and the surface underneath. What's most revealing here is that a similar treatment in adult mice, where the F-actin belts have thickened, produces no effect. I believe myosin II has less of an effect on supporting cells from adult mice than it does on those in chicks. Why is it that the F-actin belt gets so thick over the life of a mammal? And how come this has coincided with the loss of regenerative ability? It's possible that there is a logical evolutionary reason for this, but it's kind of difficult to infer with our current knowledge. I speculate that the thickened F-actin belts, which provide support for the hearing and balance apparati of the inner ear, confer heightened sensitivity for our ears. Thus, as a way to acquire better hearing, we mammals have lost the ability to regenerate after acoustic injury. Whatever the reasons, it is becoming clear that scientists might be able to induce a regenerative response even in adult mammals one day. With this comes the possibility, but not necessarily the guarantee, of a highly effective treatment option for the millions of Americans with debilitating deafness.